So, so this is about electrophoresis of DNA when we say. So basic electrophoresis, yes, it's not new to you. You people have performed also in the laboratory. So basic methodology is known to you, but here are certain highlights. Now we say DNA has got net negative charge, that is on account of the phosphorus, that is phosphate group which is present, so which imparts negative charge. That's why when electric current is applied, DNA will move towards oppositely charged, that is towards positive electrode it will move. Right now, when we talk about electrophoresis of DNA, majority of the DNA samples that we talk and its separation we want to carry out, it is done with agar gel. But it's not that ki only agar gel is useful, polyacrylamide gel also is useful, but when and how we will discuss. So why do we use agar gel? It's because the DNA molecules are, and even their fragments, even if you talk, which are routinely analyzed, they're bigger or larger than the proteins. And most of the DNA fragments, when you talk about, they are larger that are not unable, they are not able to enter the polyacrylamide gel. So the agar gel, which is having larger pore size, is required. So commonly used plasmid DNA, if you talk, say PBr322. What is the molecular mass of that? The MR for plasmid PBr322 is 2.4 into 10 raised to 6. Right. However, rather than the large number, it is having convenience to refer to the DNA size in terms of number of base pairs. Right. So originally DNA size is talked in terms of base pairs or kilo base pairs, etc. Right. But when we talk about PBRK322 in that term, then the that size in terms of kilo base pair is 4.36, right? And even a small fragment of 1 kilo base pair, if you take, its molecular mass is about 6,20,000. So when we are talking about the sieving action of the gel, the movement of analyte through the pores, the molecular mass that matters, right? So when we talk about molecular mass of PBr322, it is 2.4 into 10 raised to 6, a very big molecule, right? So when you talk about single stranded DNA, and talk about the size in terms of nucleotides, it is NT. And for DNA, we talk in terms of BP, that is base pairs, right? That's why PBr322 example I have taken, which has 4.36 kb, the molecular size, but molecular mass is about 2.4 into 10 raised to 6. So, of course, due to its negative charge, it will move towards anode with the mobility which is going to be dependent on applied current. But the size is the factor that will not allow its movement through polyacrylamide. So we are using acrylamide gels. Right. Now this is the history that I wanted to share with you of electrophoresis, just as a matter of information. There is nothing to elaborate much on. Sorry, my mic I'm handling. Okay, fine. So this is about just the history that when, as early as in 1960s, 80s, how electrophoresis started and what is the status today. You can just take a clip of it and we move ahead. Right. So when I say gel electrophoresis, we say that DNA is negatively charged, so we'll move to positively charged anode when the applied voltage is there. So smaller fragments are able to move through the gel pores easily than the big that we know, right? Now, it is very much similar or analogous to separation of proteins under polyacrylamide gel, which we have learned. Um, so, the concern here is uh, protein molecules, when they are run under page alone, their native structure is retained. And when they are run under SDS page, native structure is converted to a rod-like structure. That is the difference. But when it comes to DNA, let's take example of double-stranded DNA. Right? They are double-stranded linear DNA. When I talk, these are the long fragments, right, or long threads. So when they are going to pass through the pore, this DNA molecule will not move so easily, right? It will experience the drag because molecular size के साथ shape भी है, which is going to be important, and fragment length is the one which is going to resist its movement. Longer the molecule, more time it will take to move, more it will be retarded, right? Similarly, small DNA double stranded DNA, if you have, it will be moving faster than single stranded DNA, because single stranded DNA is the one which is a flexible strand, and it will fold upon itself, and again there will be a drag and resistance to its movement. So it is obvious that the gel concentration should be chosen such that that will allow the size range of molecules to be separated. But at the same time, you need to know what is the type of material, whether it is a dsDNA, whether it is ssDNA, or a mixture. Because if it is plasmid DNA again, it will be circularly closed ccDNA, and it will be moving fastest under the applied current in the across the gel. So shape of the DNA, shape, otherwise I shall say the structure of the DNA molecule, whether it is dsDNA, ssDNA, or it is circular DNA, accordingly its retardation or its speed up, in short its mobility will be governed. So generally gels containing 0.3% agar or gel will separate a double stranded DNA molecule. Now, when I say double stranded DNA molecule, here comes what is the size? Yes, for 0.3% agarose is used for 5 to 60 kb size DNA. But when you are talking about the sample having 3 kb DNA or zero point 0.1 kb DNA, there is 100 base pair DNA, you have to use the gel with high concentration of agarose. That is generally 2% gels are used for samples having DNA size between zero point 0.1 to 3 kb. That is 100 base pair to 3 kb, you have to use 2% gels. Routinely in practicals, you might have used 0.8% gel, which are separable, good for separating DNA molecules where the size is small, 0.5 to 10 kb. But standard 2% gels are used for samples between zero point 0.1 to 3 kb. Fine. Now, agarose gels are going to separate DNA, in short to say, according to size, the molecular mass, relative molecular mass of the DNA fragment, right? And can we determine it? Yes, we can determine it on the basis of electrophoretic mobility. Now, what is electrophoretic mobility, mu, how it is calculated? All this we have learned in the very first lecture related to electrophoresis. Right? Now, when it comes to DNA separation by electrophoresis, generally the gels are horizontal, submarine, or you can say submerged gels, the submerged in buffer. That's why they are called as submarine, right? So agarose is dissolved in gel buffer by boiling. It's poured into a glass or plastic plate. So you have the casting tray, and 3 millimeter depth gel are casted, right? Now, of course, you are going to use the uh, um, combs when you are pouring the gel so that you can generate the wells into which the samples are to be loaded. So when you are talking about loading of the mm, uh, samples into the gel, you have to take care that you are using some density enhancer, like we call glycerol or sucrose. So that is going to increase the density of the solution, and that's why the DNA when loaded into the well will settle or sink in the bottom of the well. Also, the important component is bromophenol blue, which is included in the sample solvent. So that will help to see how far is the mm, DNA has run. It is going to act as a marker for electrophoretic front. So what distance the sample has traveled, it cannot be seen unless you add the dye that is commonly used is bromophenol blue, right? Now, in case of page, we had seen the stacking gel, but commonly in when you are talking about native gel electrophoresis for DNA, no stacking gel is needed for electrophoresis of DNA, because the mobility of DNA molecule is much greater in the gel, uh, and therefore all the molecules in the well will pile up against the gel within few minutes of the current is applied. In case of protein, it was not the situation. You had to concentrate and stack the protein so that it will start running across the pores. So in polyacrylamide gel for protein, you have to have stacking gel and resolving gel. But when it is about electrophoresis of DNA, you don't need the stacking gel, right? So general purpose gels, when you talk, they are generally 25 centimeter by 12 centimeter dimensions and are run at a voltage gradient about 1.5 volts per centimeter. So these are the, of course, the size of the gel and the current applied and time, they are interrelated and they are to be optimized. I'm talking about the general purpose gel where approximately 25 centimeter by 12 centimeter are the dimensions and you are using the voltage gradient of about 1.5 volt per centimeter, but this will go on for overnight, right? When you have done it in the laboratory, you had done it for the gel which is about 10 centimeter long, and within 2 to 3 hours you can finish the gel run, right? So once the gel system has run, the DNA needs to be stained and visualized. So if you remember, you had used ethidium bromide. So the gel is rinsed gently in a solution of ETBR, or maybe you are putting ETBR in the gel when it is getting casted, right? But when you are going to wash or rinse the gel in the ETBR solution, it is generally 0.5 microgram per cubic centimeter area of the gel. That is the concentration used. So you are going to rinse the gel in ETBR 0.5 microgram per cubic centimeter concentration solution, and then you are going to take it to UV for visualization. Now, as we know, ethidium bromide is cyclic molecule, it's planar aromatic molecule, and it is intercalating molecule. So it is going to get bound to bound between the base pairs of the DNA, right? So ethidium bromide concentration builds up at the site of DNA bands, and under UV light you will see orange red fluorescence. And the efficiency of the method is such that you can visualize 1 centimeter wide bands also, and even if the concentration of DNA is as less as 10 nanograms. So efficiency is really good. Now it is very important that you should have that UV visualizer, without which you cannot see the DNA. And moreover, it is expensive in terms of experimentation, because once you add ETBR, your DNA is damaged, because dimerization, nicking, all these things, when you are exposing it to UV light, intercalation of ETBR, all these things are going to alter the DNA. So you should have non-denaturing systems for detection or visualization. You can do it with exposing the DNA solution. That is without loading it. You can just take it to the UV spectrophotometer and you can find out the concentration of DNA. But whether its stoichiometry or its properties, topographical properties, cannot be understood. Similarly, whether the structural details, like whether it is dsDNA, ssDNA, or plasmid, that also cannot be uh, understood. Neither its size can be understood. So, agarose gel electrophoresis, when we say, what is agarose? As we all know, it's a polymer. Basic unit is galactose, right? So it is polysaccharide purified from red algae. You have the anhydrogalactose units, which are linked by glycosidic bonds, right? And we can get the gelling property of agarose, and the gels are sturdy enough. So when we talk about agarose gel, how it really looks like. So this is the cross section of the structure, and the long rods that of polysaccharide units, how are they are arranged. So you can say the agarose helices are there, and they form long stiff rods. So when you go for gel surface studies using electron microscopy, the fibers of agarose freeze fracture, of course you need to do it for electron microscopic studies. So what you see is a network of rods. So agarose aggregates are there and channels are getting formed. These studies were done as early as in 1982, and they had found that for a gel of 0.25 to 4% agarose, The agarose fibers observed by electron microscopy after freeze fracture, what was the radius? Radius was 3 to 6 nanometer. So why I'm talking about this is it is the standard quantity of agarose that you are adding in known quantity of buffered caster gel to gain the desired porosity. But porosity and cross-linking of these gelling matrices are to be studied or have been studied to these details. Ki what is the morphology of the polymer that is getting generated. If you remember in the previous lecture, we have seen how acrylamide and bisacrylamide gets polymerized with the help of NNN tetramethyldiamine or with the help of photochemical decomposition in presence of riboflavin, right? So that pore size is very important. So the fibers they contain between 10 to 30 agarose helices. So this is one helix, agarose helix, two helix, three helix, like that we are talking about. And the fiber is the one which is having diameter of 3 to 6 nanometer. And these fibers are interwoven to accommodate the spaces. And those spaces are nothing but the pores through which your DNA is going to move. Right now, when it comes to mm, the type of gel that are commonly used. We know it is agar gel for DNA commonly we use, but it is generally used to separate DNA fragments from 300 to 10,000 base pairs. Higher concentration of agarose will produce a denser matrix and therefore it can be used to separate small size DNA fragments. So here the chart provides you the details about the percent of agarose and the quantity of double stranded DNA in terms of kilobase pair that can be electrophoresis using that gel. For example, the very first row it shows you the agarose percentage 0.3 and double stranded DNA size is uh, s- uh, uh, about about 50 kilobase. All right. Like that you have 2% agarose that you are using, and that means the pore size is going to be very very small. The fragments are going to be very small, so it is 3 to 0.1. So uh, s- from as less as 3 to 0.1 up to 5 to 60 kb, you have different concentrations of agarose that can be added to the buffer, and a right gel can be prepared for it. Otherwise, time, voltage, and dimension calculations would go wrong. You have to know what is the porosity of the gel, and then you can pre-calculate the time required, or you can decide upon the voltage that can be allowed without heating up of the gel and denaturation of the DNA. You have to run it to the fastest possible speed. All right. Now, when I say electrophoretic mobility of the DNA, it largely depends on molecular size of the DNA. That is one factor we have seen. Just now we talked about percentage of agarose and size of the DNA. Similarly, we have talked about the linearity of the molecule, the shape of the molecule. So linear DNA will be moving through the matrix, and it will be proportional to the its molecular mass. But how it is proportional? It is inversely proportional. So molecular weight of that or mass of the DNA and its mobility, they are inversely proportional to each other. Of course, molecular mass we don't talk in terms of 1, 2, 3, 4 or numbers as such, but we take log to the base 10 of that value so that you can span the range of it. Right. So if you have 100 base pair fragment to 1 lakh base pair fragment, right? So to accommodate on the graph, we don't talk it as 100 and 1 lakh, we say it in terms of log molecular weight of that material, and on this is going to decide its mobility. So on x-axis you have one factor and mobility on the y-axis. Of course, agarose concentration is one of these factors which is going to decide the mobility. In addition to that, there are some more factors they are to be talked about, and they include the salt concentration mainly, the age effect, and the sample contained. These three are very important, right? So when I say about these, what is your idea? Ki what salt must be doing? What is the age effect? What is the uh, 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 the effect of these factors on the mobility? Yes, anything? Any guesses?
Conductivity, ma'am.
Conductivity due to salt. All right, good point.
Yes,
Anything
ma'am.
else? So how the salt would
Mm,
affect? Will it cause what we say affect in positive way or negative way? Will
in a
the
positive way, ma'am.
like
Like it helps to flow the current. I mean,
uh,
it's increase the conductivity of the what we say, the composition or compound,
it is one aspect,
which
right? But
can,
can we think about the
yes,
viscosity of the buffer if we increase the concentration of salt? And increased viscosity will reduce the mobility.
yes,
You know, that is another factor to be taken into concentration. So at different salt concentrations, you will get different mobilities, right? This problem also comes in the picture when we talk about use of various restriction enzymes, restriction endonucleases, which are used to cut the DNA. So of course, this cutting of a DNA sample, uh, one experiment I'm just trying to share with you. It was shared, the fragments of the DNA were loaded onto the gel having different salt concentration in the buffer, having different salt concentrations. And the restriction endonucleases have different optimum salt concentrations for the activity. They are the part of reaction also. So those salts also go along with the sample into the well, and the migration of the fragments under electrophoretic process will be different, and it will be dependent on salt, because excess of salt will bring them like will cause a kind of a resistance. Excess salt will cause a kind of resistance. That is one point. And another thing that we talked about is the loading of the sample, quantity of the sample that is loaded. All right. So you have to load uh, the appropriate size of the DNA. What does it mean? If you load too much of DNA in a lane, the DNA will migrate as a broad smear rather than a sharp smear, right? And overloaded samples do not migrate far. They will travel very slowly, and you will not get uh, a good result out of it. So it is not that the size of the fragment, but the quantity of the sample that you are going to load also is going to have an impact. So DNA can be not seen as a proper precise band. It will not migrate a good distance. At the same time, you may see the trailing effect, right? So the leading edge has got of the band is there, but there is some trail remaining. So it is like smeared one on the band will be, right? Also, you have to think about the DNA sample that does not migrate at the same rate in two different parts of the same gel. Can it happen? Yes, it is observed. So differences in migration occurs because the fluctuation in the electric field maybe. So you don't have constant electric field, not through the source, but I'm talking about the electric field in different parts of the gel is differing. So how it is possible to have not to have the constant electric field throughout the gel? Different parts of the gel have different electric field. So one reason could be that the gel is getting heated up, right, when it is current is applied, and the edges of the gel which are in good contact with the buffer, they will dissipate the heat. But in the center part of the gel, the heat is not going to be so readily dissipated, and the temperature may increase at these points, and the gel in the center would be warmer. So such warm gel has effect on current, so greater current in it will have. and DNA fragments in the center will migrate more rapidly than those are on the edges. That is called as edge effect. So sometimes it gives you the effect where the aging effect results in bands which are referred to as curve bands, but they are named as smiling bands or smiling of bands, because the bands in the center of the gel have migrated farther and the gel artifacts are seen. So your gel is not going to be really the right output it is not going to give you. So this is what is referred to as smiling bands. All right. So this is overall a few things about when we talk about electrophoresis. Just uh, I think some points more than what you really might have jotted down if I would have asked you to write the answer or short note on agarogel electrophoresis. So these extra points I have tried to touch so that you can add those in the answer. Now there are two advances that we have to talk about, and they are nothing but capillary electrophoresis and pulse gel electrophoresis. All right, so pulse field gel electrophoresis. So let's talk about this. And the DNA sequencing gels, that is another thing to be talked about. But the time may not permit, so I just stick to only one of these. Okay, this is to okay, fine. This one I would take first, right? This is about the ladder and quantification of the fragment size. So, as an application, when we talk about separation of DNA using electrophoresis, so separation of differently sized fragments can be easily done. That is what we write. At the same time, we talk about the ladder, right? And the application of this method that you can very well write is determining the relative molecular mass of unknown DNA fragment using a DNA ladder. So, what actually is the relationship here? If you see, these are the relative mobility, the distance traveled on the gel. Against that, you have plotted log molecular weight of the DNA fragment, right? Or that of the bases in that fragment. So, more the log molecular weight, smallest is or lowest is the mobility, and vice versa. The highest mobile um, fragment is going to have least molecular mass. So by plotting such graph in comparison with um, the fragments having known molecular mass and their mobility, you can plot the graph, and then that of unknown can be the molecular mass of unknown fragment or the fragment under study can be determined. So size based separation, of course, that is what we achieve uh, uh, when we talk about the fragments. Of course, as I said earlier, the structure, whether it is dsDNA, ssDNA, or circular DNA, that is going to be important. But yes, appropriate sizing, migration distance of the sample, they can be compared to migration distances of molecule of known size, that is molecular weight standards. And these standards, when put together and they run on the gel, we call them as, they form the ladder. But this is one of the applications, that is this molecular mass of the ladders components are known, the migration distance you can measure and plot a graph like this, and the molecular mass of your fragment under study can be very well correlated. Molecular mass and fragment size and the distance travel mobility, they are interrelated, so you can very well mm, talk about it, right? So you can define it. Now here is the schematic diagram. It is little blurred, but yes, these are the sequencing gels. So Sanger sequencing, when we talk, we have learned it in previous lecture in systematics, right? So you have special gels which are required for separation of DNA fragments when we are using DNA sequences. So they are referred to as DNA sequencing gels. So, whichever DNA sequencing method is used, the final analysis you have to use the separation of DNA molecules and single stranded DNA molecules. And of course, the shortest size that is used in sequencing by Sanger is, it can be as high as 1000 nucleotide. The smallest fragment that is sequenced is by Sanger's method. By NGS, you can sequence the entire genome, but of course, there also you have to go for fragmentation. So the single standard fragment about 1000 nucleotide, and you have to separate. So if there is a difference of single base, then again, on the basis of size, you can separate them out, right? So whenever you are talking about DNA sequencing gels, it is necessary to have a small pore gel, and there we use acrylamide gels, right? We do not use agar gel for DNA sequencing. Generally, 3.5% polyacrylamide gels are used for DNA, which is in the range of say 80 to 1000 uh, nucleotide single strand है यहाँ पर. That's why 100 to 1000 you can remember. Polyacrylamide gel is 3.5%. And if your DNA is between less than 100 nucleotides, what to be done? Then you have to use 12% polyacrylamide gel. So a wide range of sizes are have been analyzed using polyacrylamide gel, and of course you can use a gradient gel also, right? So in the gradient gel, it is not about temperature like TGG, it is not about denaturing gel like DGG, but I'm talking about gradient in terms of quantity of polyacrylamide. So maybe the gel is having different zones having different concentrations of polyacrylamide, maybe from 3.5% to 7.5%. That is how the gel is. So at one end it is as less as 3.5%, and another end of the gel is having concentration about 7.5% of polyacrylamide. So sequencing gels are run in the presence of denaturing agents, but the concentration of polyacrylamide also is varying. So denaturing agents, as like in DGG technique, urea or formamide can be used. Of course, this is required to separate the DNA molecules that are very similar in size. So the sequencing gels, which are to be very long, they are not going to be 10 centimeter like you have done it in the laboratory. The sequencing gels are about 100 centimeters size, so that good separation can be achieved, right? So electrophoresis in agarose can be used for preparative method of DNA, but when it comes to analysis in terms of sequencing of gel, then you have to go with polyacrylamide gels. Of course, any sequencing method has to rely on DNA sequencing gels. So DNA sequencing gels, what are the highlights? It's not agarose, it is polyacrylamide. It can separate the DNA fragments which differ by single nucleotide length size, right? That can be separated. Third point, you are going to have different types of polyacrylamide gels based on the concentration used. If the concentration is between 20 to 100 nucleotides, then you can use 12% polyacrylamide gels. And for higher quantities, you can use 3.5% acrylamide gels up to 1000 nucleotides. And last point was that you may have to go with gradient gel. At one end, you have 3.5%, another end, you have higher concentration. The pore size is small, and small fragments can be held there or positioned there or stacked there or will form the zones or bands, right? So these are the some of the summary points for DNA sequencing gels. A very small point just to touch, probably it is not there in the syllabus, but yes, as an advancement and applicability, considering that you must be having basic information about that.